Hey guys, welcome back. So we've had a little break and now we're going to get back to it by learning a little bit more about DNA and DNA replication. So before we get into the mechanics of DNA replication, I think it's always important to understand sort of the historical aspect of how DNA was really found to be the genetic material. So to set the scene, we can kind of think about what scientists already knew around the time they started to discover that DNA was, in fact, the genetic material, the source of heredity. So people really knew about inheritance. They knew that if you had a kid, then your kid might look like you. They sort of understood principles of inheritance, and DNA had already been discovered as a molecule. So this is a key point. People already knew about DNA. So this was actually discovered well before some of the events that we're going to be talking about in the 1800s. People knew that DNA was there. They knew more or less its composition in terms of having this sugar phosphate backbone. They sort of didn't really know about the backbone, but they knew that it's got some phosphorus in it. It's got some nitrogenous stuff going on. They sort of understood the ingredients. So it's kind of like if you looked at the back of some type of snack, whatever your favorite snack is, looked at the ingredients, you might not know that that was your favorite snack just from looking, but you sort of would know what was in it. That's kind of the understanding that we had about DNA at the time. So we're going to be really focusing in this unit on DNA being the genetic material. So not just some random molecule that we can play around with, that we can, determine the composition of. How did we get from the discovery of DNA as a molecule to DNA as a genetic material, and then finally to the structure of DNA? So we're going to be talking about several experiments and scientists that contributed to this knowledge. So Frederick Griffith, of course, will be talking about helping us determine that DNA is the genetic material. So none of these folks that we're talking about today discovered DNA as a molecule. That was actually Frederick Griffith, which we'll talk more about in molecular genetics if you take that course. So Frederick Griffith is going to help us determine that DNA is the genetic material. Experiments by Avery, McCleod, McCarty are going to help us confirm that. We're going to see Hershey and Chase as well. Okay, Chargaff also contributing to the structure of DNA. Rosalind, Franklin, of course, and Watson and Crick, kind of. Okay, so connecting these points, they were all kind of thought about separately, and eventually we figured out that DNA is a molecule, but it's very important. We go to figure out that it's the genetic material, and then the structure of DNA, and then we'll talk about how we understand it to replicate. So how do we know that DNA is a genetic material, and what is the structure? So these are going to be the experiments that we're talking about today, kind of in order so you can think about the way that scientists would have thought about these kinds of problems as they solve them. Okay, so let's start with Frederick Griffith. So I'm sure you guys have heard about Frederick Griffith before, but for some of you, it may have been a little while. Some of you may have not heard about it, so we're going to go ahead and talk about it. And it's always important not just to memorize what contributions these scientists made, but to really think about the mindset of the science. What problems were we trying to solve, and how did these scientists get information? Okay, so Griffith used a couple of different cells, specifically 2 strains of bacteria, one that is going to be called S for smooth. Okay, so you can see that it's got kind of a smooth sort of coat, and one that we call R for rough, and this is going to be named after what we see when we look at them under the microscope. Okay, so these are very similar strains of bacteria, but one is going to produce a smooth coat, the other is going to be left rough. So of course, as scientists do, we inject things just into mice, and it turns out that these S cells give our mouse pneumonia. So if you give a mouse pneumonia, okay, this mouse will die. All right, so the S cells are pathogenic. They cause illness and potentially death in these mice, okay? Whereas the R cells, which again, they're a very similar strain, you give these to the mice, they stay healthy, okay? So it turns out, a little side fact, that this has to do with the immune system's perception of these two different types of cells. The S cells, the smooth coating kind of makes it easier to evade the immune system. Okay, but that's not really the take home here. That's just to help you kind of remember what happens. Okay, so that's all fine. We have, this is not the experiment, this is just the setup. Okay, so next what we're going to do is kill our S cells using heat. Okay, so we've heat killed these S cells, and you guys know this is why we cook food, to prevent bacterial growth, to kill anything that is on it. That is why we, heat things to sterilize them. We kill these bacteria. Okay, these should not be able to cause disease. Okay, and in fact, giving dead cells to the mouse does not cause pneumonia. Okay, so far, here's what's going on. Okay, this all seems like a natural process. So let's check out what happens if we take these same dead cells. Okay, so they are dead, they did not cause disease. We're going to take them, we're going to mix these with living R cells. Okay, so in our next experiment, that's what we mix together. So R cells do not cause disease. Dead S cells do not cause disease. However, in this equation, no disease plus no disease equals dead mice. Okay, and if your mind feels blown right now, imagine being Frederick Griffith, who's like, what in the heck is happening right now? Okay, so I gave them no death plus no death equals death. And even more interestingly, living S cells could be removed from these mice. So not only did we kill all these S cells, we recover them alive. So are they zombies? What's happening? Okay, so this had to be like really, really just mind-blowing at the time. Okay, so we kind of take it for granted now, but put yourself in this position. Okay, so what is actually happening? What the heck is going on? So let's kind of take a closer look at these bacteria in this mixed dead S and living R sample. Okay, so important to remember that these bacterial cells have DNA. So circular DNA, because they're prokaryotes, so I am showing their circular DNA here. Okay, and here what I'm showing you is something that Frederick Griffith didn't know right at this moment, but it's what's happening. Okay, so it explains what's going on. So while Griffith didn't know this at the time, what happened is the genetic material, the DNA from these dead cells, actually was taken up by the living cells. So whatever it was that made these S cells produce this smooth coat and make them deadly, they have transferred that information in the form of DNA, which again, Frederick Griffith did not know this. Frederick Griffith knew that if you give this mixture to mice, they die and living S cells come back. That is what Frederick Griffith knew. We know in the future, in 2020, that the DNA is being moved. Okay, so that's what's happening, and then that causes them to produce this smooth coat, which allows them to convert to the S strain. Okay, so again, what he knew at the time is just that he was able to kind of regenerate these S cells. Okay, and so his idea from what happened, his take home is some information is being passed from the dead cells to the living cells that help the living cells become disease causing. Okay, we call this horizontal transfer because it's not from parent to offspring. It's from like your dead friend giving you something that you turn into your friend. Okay, so this is horizontal. Okay, he did not know for sure what caused it. Okay, this process is called transformation. So the R cells were transformed. Okay, so this is transformation, and when we get into sort of modern times, transformation we'll use anytime we're going to use bacteria to take up sort of extracellular, extra DNA. Okay, so this is what we know. And our take home so far is that information passes from one cell to another. So this is kind of an interesting take. People obviously sort of realize that some essence from you passes to your offspring when you have them. But this kind of illustrates, wait a second, this is like a real information that we might be able to find. This isn't just magic, parent to offspring. This isn't, you know, their eternal bond of love. This is a concrete piece of information that we might be able to dig around in here and find out what it is. Okay, so that leaves us to the next experiment. Okay, so how was the source of the information, which we know to be DNA here in the future, figured out? So that turns out that experiments by Avery, McCleod, and McCarty are going to be sort of the thing that first tells us that DNA is that information. Okay, so just to set this up a little bit further, at the time, people very much liked biochemistry. So thinking about sort of the context of sort of like feel that scientists had, people really thought that amino acids were probably the hereditary material. So that's right, proteins. They thought this because they already knew that there were 20 main amino acids, and they're like, this can give us diversity. Whereas they understood ATCG, and they're like, that's not enough. 4 is not enough to give us the diversity that we need. So we're kind of thinking, okay, we got to tell the difference. Like, is it protein that's being transferred over, or what? What's happening? So that leads us to their experiments. So the first thing we do is take those dead S cells. Okay, so they are dead. They should not be causing any problems here. Okay, and what we're going to try and do is figure out under what circumstances we can get living S cells sort of reclaimed. Okay, how can we figure out if transformation is going to happen? So we take those heat-killed S cells and sort of break them open so that all of their juices spill out together. So this is the combined essence of all of those S cells. And we have several different types of molecules. Okay. So there are 4 main types of biological molecules: carbohydrates, okay, that we're going to be dealing with here, which we're going to remove. All right. And actually, maybe we'll count lipids too, since we've got them here. We're going to take those out, okay, because we already sort of know that these are not really going to do anything. We're pretty sure about that. So that leaves us with our 3 remaining types, okay? So first, the dead S cells, again, were lysed. Their molecules mixed together. We remove carbohydrates and lipids so that we are left with proteins, RNA, okay, so there's that, and DNA. So again, people knew these molecules existed, but they weren't sure which one of them could be the source of information that can be passed on. Okay, so the next thing that we need to do, and you can tell we're about to do something in these 3 tubes, okay, is we're going to systematically destroy each of those 3 molecules one at a time, proteins, RNA, and DNA, systematically destroy them in these samples, and leave the remaining molecules and see when we can still transform. Okay, so let's get started. In our first sample, we add proteinases. Proteinases are a general type of enzyme that destroy proteins. So there are no proteins in this sample. There is still DNA and there is still RNA. Okay, next we add ribonuclease, like ribonucleic acid, like RNA. So this is ribonuclease. This will destroy RNA. So this sample will contain no RNA. It will still contain DNA and proteins. Okay. In our last sample, we add deoxyribonuclease, or again, DNase. So this will destroy DNA. So we have a sample where we remove the protein, we've left RNA and DNA, which is assuming the DNA is red here. In our next sample, we've destroyed RNA, so we still have protein and DNA. And in the third sample, we have destroyed our DNA, but we still have our protein and RNA. Okay, so in 2 of the samples, we still have DNA. In 1 sample, we do not. Okay, so we take those samples and we mix them with living R cells. Okay, so again, what we're going to try and do is get that same transformation that Griffith observed to happen in this situation, but we've systematically destroyed molecules to see which one is causing the transformation. So mix the molecules with our living R cells, and hopefully you already know the answer to this, but in samples that still contain DNA, which is red here, we are going to be able to transform because the DNA is what's giving that information. Okay, S cells appear. Here, again, we've destroyed the RNA, but the DNA is still intact. And man, there are those S cells. No DNA, no S cells. So that told these guys, hey, DNA looks like it's the information. It's the information, man. Okay, so that is one of the first experiments that's going to tell us exactly what's happening here. Okay, so that's going to tell us exactly what's happening. All right, but we're scientists, so we're going to have to redo this. Okay, we're going to have to do it in another way to confirm. All right, so we know now that DNA is a hereditary material, but we're going to confirm it with the Hershey and Chase experiments. Okay, so you guys again may have heard about this one as well. So Hershey and Chase find out that DNA is the genetic material using a completely different experimental approach. So to do this, they use a bacteriophage called a T2. To understand how this experiment's going to work and why they use this bacteriophage, we have to learn a little bit about how these guys operate. So bacteriophages are made of protein, kind of coats and shells, little legs. These are all proteins. There are no lipids, no carbohydrates, nothing really going on here. This is protein and DNA, man. That is it. It is a simple being. Okay, so, and you can also see in this animation, the protein shell, the DNA was encased up here in the top. The protein shell lands on the surface of this bacteria, and it's going to inject its DNA. Okay, so that's going to give us some really good setup for the next set of details that we're going to learn about this experiment. So there's simple structure, only protein and DNA. And the way that they inject this material is going to make it really clear that DNA is going to be the genetic material. Okay, so let's check it out. Their experimental setup. All right, so basically what they wanted to do is kind of differentiate where the DNA and where the proteins are in these E. coli. So once the bacteriophages get done with them, again, they didn't know, okay, man, these are injecting their DNA and that's what's causing the infection of these bacterial cells. They just knew this is the way a phage looks. This is more or less what it does. Let's find out sort of what's going on here, okay? So we'll figure out if it's DNA or protein based off of where these are in the bacteria, okay? So ostensibly, if we're going to be making changes to these bacteria, the item that is going to give the information will be injected inside of it or left inside of it, okay? So what we're looking for at the end of the day is what is inside these bacteria. Is it DNA or is it protein? So let's check it out. So to differentiate between the DNA and protein, which is the first thing they have to do, they have to be able to tell where the DNA and protein are. They have a set of samples that they grow with radioactive sulfur. So the radioactive sulfur, we're labeling in pink here. This is important because we're going to label our protein. So we'll see that in this next slide, okay? The second set of samples is incubated with radioactive phosphorus. So phosphorus is going to help us label our DNA. So these are now labeled separately, but how? So proteins contain sulfur. These are going to be present, these sulfur atoms, in certain amino acids, which we know are the building blocks of proteins. Particularly, I'm showing methionine here. So proteins contain sulfur, while DNA does not. DNA, however, does contain phosphorus in our sugar phosphate backbone. So what we're going to be looking for is, again, labeling our proteins versus our DNA. Okay, so we've labeled one versus the other. All right, and remember, just to think about it from their perspective, they're not seeing all this jazz that's going on with these E. coli and what's going on here. They're seeing radioactive sulfur and radioactive phosphorus. Okay, so they're just going to figure out where this radioactivity goes. If they see radioactive DNA somewhere, they'll know where that went. If they see radioactive protein somewhere, they'll know where that went. Okay, so on to the next step. Once we've grown these phages and E. coli together, we're going to sort of disrupt them mechanically, so kind of shake them around in this blender. And the point of that is to sort of dislodge all of the external pieces off of the E. coli. Okay, so we don't want to get confused. If something's stuck on the outside, we don't want to confuse that with it being injected inside. Okay, we don't want it to be mistaken just because it's there together, stuck physically together. We got to knock those guys off. Okay, and again, we can see sort of the spoiler. We know that the empty protein shell gets knocked off while the DNA stays inside, but we got to think about it from their perspective. Okay, so the next thing they did was a differential type of centrifugation in a way. So this is very similar to what you guys learned about before. But really what we're trying to get, we're not trying to isolate anything small relative to the bacterial cells. We're not trying to get ribosomes or anything. All we're trying to do is pull the bacteria to the bottom of this tube and leave the supernatant. So we're trying to answer the question, okay, if the item, that is the information here, goes into the bacteria, then that item will be with the bacteria no matter where it goes, okay? So we're trying to separate the bacteria from the liquid that they grow in, okay? So here we can already see our protein has been labeled pink, and again, this is what they're looking at, okay? They're not looking at all these phages in business. They're looking at these tubes. They're going to pull some of this supernatant and see if it's radioactive, and they're going to pull some of this pellet and see if it's radioactive. They'll do the same thing with their radioactive phosphorus treated samples, okay? Centrifuge them, get the pellet. The bacteria are going to tell us what's going on. Check them for radioactivity. So it turns out in the sample where the proteins are labeled, the supernatant is radioactive. That's all those shells that got knocked off here. The supernatant is radioactive while the pellet is not. That means that proteins are not inside the bacteria, okay, at least from the phages. Okay, they have their own proteins, not phage proteins. Okay, proteins are in the supernatant. In the sample where the DNA is labeled, the pellet is radioactive. Okay, so this means that the bacteria themselves, whatever's inside them, okay, that radioactive item is in there. Okay, so that means DNA is inside these bacteria. That is confirmation that that's the information, because these phages are so simple. They're just proteins and DNA. So it's one or the other. If the proteins aren't even inside the cells, gotta be DNA, man. And this is cool because we're really working with various organisms. Okay, we had bacteria that we're looking at in the Frederick Griffith, the Avery, McCleod, McCarty experiments. We're now looking at a phage. Okay, it's pretty much the simplest thing that may or may not be alive. We can talk about, these are two different types of things. They're both pointing to DNA being the genetic material. Okay, so we can kind of remove these spoilers and see what happened. Again, we've disrupted these phages off the outside, and the DNA is left inside We've labeled it in these blue samples, so that's why we can see it. The DNA is still inside these bacteria, but we have not labeled it, so we just can't see it. Okay, so that's the Hershey and Chase experiment. This is a really cool experiment, you guys. This is just really beautiful in a lot of ways. Okay, so thinking about, again, we take this stuff for granted. They thought about this and designed it in such a really just excellent way. Okay, so this is a cool experiment. All right, so we're getting closer. We've gone through the experiments where we're like, okay, we went from DNA is just a molecule that we sort of knew about to, okay, this is the important molecule. This is where people really started to get in that race for figuring out the structure of DNA, because now we know this is the important thing that we're needing to go after. Okay, this is what we need to figure out about. So we know about the molecule itself and sort of the ingredients. Again, we knew enough about it to know that if we put phosphorus in here, we're going to label it. And now we know it's the important guy. Okay, so let's start getting into the structure. So we'll start out by some experiments that were done a little bit separately. Okay, these were done before we really start getting into necessarily the race of DNA and its structure. But again, thinking about, oh, DNA is a molecule, characterizing that molecule. Okay, so we knew that it had 4 different things. We've got adenine, guanine, cytosine, and thymine going on. Okay, so Erwin Chergaff is actually going to really quantify the amounts of each of those nucleotides. Okay, so he takes a lot of different organisms. We got some phages, got that E. coli again, and Neurospora. Okay, so that's fungus. We've got plants, corn. We've got even up to like insects, octopus, human. Okay, man, we got the whole gamut here of different types of things. Okay, and we figured out how many of each nucleotide is present in their DNA. Okay, so what we figured out here is that most of the time, all right, what we're looking at is kind of a 1-to-1 ratio. And yes, they're not exactly 1, that's how science is, but more or less a 1-to-1 ratio of A to T. Okay, so the amount of our adenine is the same as our amount of thymine. Okay, so if we look at, let's say, this sample, our E. coli, 23.8% adenine, 23.1% thymine. So that's going to be kind of a 1-to-1 ratio. Okay. Our GC content, guanine to cytosine, which we know now in the future that these base pair together, about equivalent. And these ratios more or less hold true all the way down. And you can see, like, let's check out tetrahymena. It's not just that each of these get 25, 25, 25, 25. Look at this. This is really low GC content. So there's a lot less guanine and cytosine than there is adenine and thymine, but still, adenine is the same as thymine and guanine is the same as cytosine. Okay, so that's important. It's good to look at lots of organisms because this helps illustrate our point. So what we get from this is that adenine has some kind of relationship with thymine where they're equal, and similarly cytosine has some kind of relationship with guanine where they're equal. Okay, so this is going to ultimately help us understand base pairing rules that are coming up. Okay, so now we're really getting into the structure. So we've got some things hinting at it. Okay, it took a long time, okay, to go from, okay, DNA is a molecule, it's an important molecule, to, okay, here's the structure. Okay, this wasn't a 3-hour lab. This was like a lot of people's life work. Okay, so Rosalind Franklin, of course, we can't talk about DNA without talking about her. Okay, and her claim to fame that everybody knows about is this famous X-ray crystallography, this X-ray diffraction photo of DNA. Okay, and if you can't look at this and tell what's DNA here, that's okay. This requires some calculations in math, and you got to know some of the angles here. You don't need to know how to read this. But the important thing is that prior to Rosalind Franklin's work, there were several hypotheses about how we're really regarding the structure of DNA. So the famous Linus Pauling triple helix that we were considering at that time. Okay, so there are a lot of different things that we were thinking about. We sort of actually knew it was helical, but are there 3? Are there a dozen? Like, well, how many are in here? Okay, so this provided definitive evidence that this is a 2-strand double helix. Okay, so I think that's what you can glean from this. And Rosalind Franklin knew that this was a double helix. This was her work. Okay, but she didn't immediately connect. You know, this was not immediately connected to the full-on structure of DNA. So it took Watson and Crick taking a little peeky peek at her data and some other people's data to really get that structure. But this was one of the key pieces. Okay, so let's get to Watson and Crick. All right, so I don't know what kind of like meme situation you guys have. I'm gonna leave this up here, and if you know what this is, you're an awesome person. If you don't, I'd do a Google, man, because this is all I think about when I think about Watson and Crick. Okay, because they pretty much came in, did zero data, zero work themselves, real smart individuals, took all these pieces from different people that they talked to, and they went ahead and figured out the structure of DNA. Okay, so all your base belong to us. All right, if you're into badly translated video games, then this is, again, worth a Google to understand, but this is how I think of Watson and Crick. Okay, so what did they determine? Okay, we've talked about the structure of DNA in this class previously, and I am willing to bet many times before this moment, but let's just review. I think it'll be good before we get into the mechanics of replication. So of course, the double helix, 2 strands, as Rosalind Franklin's data indicate. Okay, we have that Chargaff base pairing ratio thing going on. Okay, we're going to pair these together. That's where that came into play. G to C, A to T, T to A. All right, and what is happening between this? These are hydrogen bonds, and you may note that there are 2 hydrogen bonds between A and T. There are 3 between C and G. It's not something we're going to focus on really in depth at this moment. It does have implications for the biology when you really get down into some of the stuff, but for right now, we're just going to kind of make note of it. Okay, notice these backbones. Okay, so we can see the sugar, so this is going to be deoxyribose. Okay, so that's deoxyribose, that's a sugar. All right, so it's going to have our 5 carbons, 1, 2, 3, 4, 5. Okay, so 3 is here and 5 is here. Okay, so got that sugar. We're going to have phosphates between them. That's the sugar phosphate. Okay, and these are connected through what are termed phosphodiester bonds. Okay, so you'll need to know that term. We're not going to look heavily into the chemistry. Okay, let's make note, anti-parallel, my friends. Okay, so check it out. One of these sides is running from the top. This is the 5th carbon, 5 prime to 3 prime. Okay, so here's the 3 prime of this guy. 5 prime to 3 prime. All right, so there we go. 5 prime at the top because this 5th carbon is upwards. 3 prime at the bottom because this ends in that 3rd carbon of this sugar. Okay, anti-parallel. Okay, so the 2nd strand runs in the opposite direction. This is critical that you understand this concept coming up to the mechanics of replication. So notice this one's 3rd carbon is at the top this time. And if we come down here to the end, this one's 5th carbon is at the bottom. This one is running 3 prime to 5 prime. So most things that we're going to talk about kind of prefer a 5 prime to 3 prime thing. I think of this as like, look at these oxygens. It's an arrow pointing up. This one's running 5 prime to 3 prime. This one, the arrow's running down. We start with a 3 prime at the top, end with a 5 prime. Okay, so I think it's important to eventually get accustomed to knowing the 5' 3' thing in terms of carbons and what's actually happening. But it's imperative that you understand what this is going to be when it's anti-parallel. Okay, 5' to 3' on one strand, 3' to 5' on the other. It's going to make replication so much easier if you get that solidified. Okay, so the structure of DNA, because of the way that it is with two strands and these sort of matching bases, provided some really good clues about how this might replicate. So even Watson and Crick sort of, from looking at the structure, predicted that because of these base pairing things that are going on, you could actually use one of these as a template while the other one is going to be added on or synthesized. Okay, so they hypothesized this, and then Messelson and Stahl came in and showed what's going on here. So DNA is replicated via what's called the semi-conservative replication. Okay, so this is our parent molecule, simplified version of what we saw on the previous slide. These strands separate, okay, they just come apart. This is the hydrogen bonds coming apart. That's all that's happening. And for each of these, we'll call them the parent strands because they were together before the new strands appeared. Each new strand is synthesized using the parent strand as a template. So what we're going to see is that go along and we're like, okay, an A was here, that means I need to put a T. If this is a C, I'll put a G with it. If this is a T, I'll put an A with it. So the light blue represents a newly synthesized strand. So the two parent strands come apart and each one has a newly synthesized partner that was created based off of complementing its own sequence. Okay, this is called semi-conservative. Okay, so again, this was determined by experiments performed by Messelson and Stahl. Okay, so let's talk about the different ways that DNA could have potentially replicated and why this turned out to be semi-conservative. Okay, so let's take the conservative model. Okay, here we're denoting the parent strands as being a darker blue throughout this sort of model. Okay, so if the conservative model was what's happening, okay, let's remember that it isn't. This is a hypothesis that wasn't correct. Okay, this parent cell that's going to have dark blue parent strands, in the conservative model, we retain that partnership, okay, no matter what, through all generations, first replication, second replication, and so on, okay. The newly synthesized strands are together, okay, so check it out. We synthesize a new strand, we keep our parent strand. In the second replication, this new strand gives rise to two more new strands where ostensibly this guy, or one of them, is exactly identical to what was going on here. The other has two newly synthesized strands. Here are the two parent strands. Here they go. Okay, so when you get done with this video, I would recommend taking a little bit of time to think about this to make sure, because there's a lot of jargon going on here and it can get confusing. Okay, but let's move on to the semi-conservative model. This is what's actually happening. This was the correct hypothesis. Okay, here the two parent strands come apart. Those are the dark blue ones. And a new daughter strand is synthesized for each for its partner. Okay, so in our next replication, there's that original parent strand that started way back here. It's going to get an even newer partner. This new-ish partner from replication 1 is now maybe this strand here, and it got a brand new partner as well. Okay, similarly, that's what happened at the top. Okay, so for right now, we're just learning about conceptually what these mean. We have one more model to go through, but we'll be kind of learning what this means and how we can tell these apart to even figure out which of these types of replication is going on. Okay, for our last model, which is the dispersive model, take our parent strand, okay? Notice what's happened in the first replication. Okay, so there's kind of a mixture here, a hybrid, where here's a chunk, both strands at the same time in this chunk are from the parent, but in this neighboring chunk, these are all new, both strands. Okay, light blue means new. All right. Similarly, we got chunks of old and chunks of new kind of horizontally dispersed. This is the dispersive model. Okay, so let's think about what that means for the next generation. In principle, each one of these two strands is like a 50-50 blend of parent and new. That's similar to our semi-conservative model. It's a 50-50 blend, parent and new, but it's kind of allocated differently. Okay, this is one whole strand and one whole strand. This is like both strands at the same time. So that has ramifications for the second replication, which is why we're even talking about it in the first place. Check it out. This darker blue, while it will stay paired together, okay, will become a smaller and smaller fraction of the overall whole. So if this is 100% parent, each one of these is 50% parent, each one of these is 25% parent, okay, and then we'll get to like 12.5% parent if we keep going until it's completely kind of diluted out. With our semi-conservative model, note somewhere in this, there's always couple of molecules that have one parent strand and one new strand. That parent strand stays intact the entire time. We're not mixing it up. Okay, so that's going to have ramifications for the next experiment that we're talking about, okay? What we've done now is not even started the experiment. We've only talked about their hypotheses, which is very important to do before you just go and start labeling stuff and making it radioactive and like all these things, okay? So we're not going to deal with radioactivity so much. What we're going to be dealing with here are isotopes of nitrogen, okay? So we've got regular standard nitrogen, okay, N14 or light nitrogen. You can think about light nitrogen as being lighter in color. N15 is heavy nitrogen, so it's just a little heavier, okay, than our regular nitrogen, and that's going to be a darker blue. Okay, this is going to work because heavier nitrogen, this is why we had to learn about centrifugation early on. Heavier nitrogen will go further towards the bottom of the tube, okay, whereas lighter will stay near the top. Okay, and I'm going to give you a little bit of a spoiler. These are the actual results that Messelson and Stahl collected. Okay, so this is going to tell us it's going to be semi-conservative, but we're going to go through all of these things together. Okay, so these experiments, we're going to use heavy and regular nitrogen, and as these bacteria in these samples replicate, they're going to be creating new nucleotides. That's the thing. We're creating new DNA as we go. So if you've been living in this sort of, you know, broth, this sort of soup, where the only nitrogen available to you is heavy, you're going to use that to make your nucleotides. Similarly, you get transferred to our light nitrogen, you're going to start using that instead. Okay, so if we start you in heavy, move you to light, we're going to be able to tell the difference between old and new strands. So that's the key, and that's how everything was labeled previously. So whatever's in there, we're going to use it, and then we're going to be able to centrifuge it to tell what's going to actually be the composition of this DNA. Okay, how much is old and how much is new. So you can kind of think about it as we go through these things, is the older will be closer to the bottom, the old strand will be close to the bottom, the newer strands will be closer to the top. Okay, so that's kind of the thing that you could say to yourself. So let's look at the results that we should expect So not necessarily that we got, but that we should expect for each of these models. So let's start with the conservative model, where again, the idea there was parent strand always stays with parent strand, always stays with parent strand. We've moved these guys to light nitrogen. Everything new is synthesized with lighter nitrogen. Keep that in your mind. Old is heavier, new is lighter. Okay, so again, we hypothesize that one of these strands, the first replication, will be entirely in 15, entirely in 14. Then we'll have our N15 strand retained indefinitely. So let's look at what we should have seen. And so we've got this big X because we already know it's semi-conservative, but ignore the giant X and let's look in the background. Heavy parent strand, all right, our lighter new strands. Check it out, that heavy strand stays there, okay? If you did a third replication, it would stay there, okay? It just will be there forever because that is what happens. Let's check out the semi-conservative model, which we know to be the truth. Okay, so this is our semi-conservative model. We have two strands, okay? We know that the parent strands came apart. We synthesized a new one using the old one as a template, okay? So that's what we did, which means in the first replication, the DNA should be a N14, N15 hybrid, which should make it an intermediate weight, half heavy, half light. You're like a 14.5, okay? So you're between 14 and 15 total weight in the middle, okay? So second replication, you can see we still retain our parent strand by itself up here. It will come apart again from this new strand, create a newer new strand, okay? Similarly down here. Look, these guys that still retained one of the parent strands will be found here in the middle. Now we have lighter strands, okay? And again, this is what Messelson Stahl actually got. So that's what we should see, okay? Let's check out the dispersive model. So if you remember this one, it's kind of a parent strand, daughter strand, 50-50 blend, but in sort of a vertical way, not necessarily like one strand and one strand. And if you remember, we said this is half heavy, half light, okay? So we're hypothesizing that the parents will blend equally, half heavy, half light, okay? That's what we get, okay, if we are going to have the dispersive model. Note that we can't tell the difference between these two things in our first replication. That's why we had to have the second one, okay? Because this doesn't know what the actual sort of structure is. This knows half heavy, one strand, half light, other strand, half heavy, parts of all the strands, and half light, parts, both strands, okay? So it doesn't know, it knows 50-50, intermediate weight. Okay, so what should we expect going into the second replication? Okay, so the N15, the heavy nitrogen, will be diluted in all the subsequent things. So take note of where this band actually is in this centrifugation experiment, okay? This is gonna be not quite at the top, okay? So notice that these lighter ones are completely at the top. This is a little further down, but it's not all the way the middle down. This is what you get when you have about 25% of the heavy and 75% light. You're not all the way up here, okay? So think about what do you think might happen if we allow these replications to continue. Okay, so in this section, we've talked about sort of the history of DNA. We've talked about how we got from, okay, this is the molecule we know about, to this is the one that is important for hereditary information, to this is its structure, and now we know more about how it replicates. So in the next video, we'll be looking at the actual specific mechanics of replication.